Thank you everyone for joining again the live lecture, or if you're watching them later, the recording. So first I want to start by summarizing a little bit covered in the past lecture. And so we learned about clonal selection and expansion, which occurs after a naive lymphocyte is activated, and then it will proliferate and differentiate into an effector lymphocytes, and only the lymphocytes, the naive lymphocytes that are engaged by their specific antigens are the ones that will be clonally selected, right? Because are the ones you want to make more copies of, because that will be used by the pathogen. So we learned that there are two major types of adaptive immune cells: the B cells that mediate the antibody immunity or humoral immunity, and the T cells that mediate the cellular immunity. And we learned that they recognize antigens in a slightly different ways. The B cells recognize antigens through the BCR that binds to native antigens, okay? They don't processed into peptides, they bind to native antigens, and then they release the soluble form of this BCR, which is the antibodies that can neutralize, opsonize, or fix complements to fight the pathogen. And we learned that T cells recognize the antigens through the T cell receptor, TCR, and when the antigen is presented as a peptide in the MHC class molecules. And these MHC class molecules can be expressed ubiquitously, like the MHC class 1 molecule that is expressed in all nucleated cells, or can be only expressed in APCs, like the MHC class 2. So the helper CD4 T cells recognize antigens presented in MHC class 2. The killer CD8 T cells recognize antigens presented in MHC class 1. We learn about dendritic cells that link innate and adaptive immunity and are professional antigen-presenting cells because they have constitutive expression of co-stimulatory molecules that give a positive signals to the T cells and are necessary always to initiate the activation of the naive T cells. Without them, we cannot initiate the activation of naive T cells, and that's why they are called the most professional antigen-presenting cells. And so we also learn about the functional outcomes of effector lymphocytes. We already said that for B cells, they will provide antibodies that neutralize, opsonize, or fix complement. And for T cells, the CD4 T cells will help other cells like macrophages or B cells that present their specific antigens via MHC class 2 peptide. And for CD8 T cells, the infected cells or tumor cells that present their specific peptide antigens in the context of MHC class 1. And then we learn the interrelationship between innate and adaptive immunity, that innate immunity is necessary for influencing the type of adaptive immunity because it will promote production of cytokines, for example, by dendritic cells that will influence the type of adaptive immunity that will be generated. And also the adaptive immunity will empower the innate immunity and will also have direct mechanisms to destroy the pathogens like the killing by CD8 T cells. And last, we started talking about where is our immune system. And so we talk about the primary lymphoid organs and that are important for development, and the secondary lymphoid organs that are important as meeting plates between pathogen and antigen and T cells. And so, and B cells as well. And so we'll learn a little bit more about this today. Okay. So with that, I would like to remind you that in the course website, I have uploaded several very short movies, but I think it can provide you a perspective and help you illustrate some of the concepts that we are learning in this course. So these are some of the ones that I think that you will find useful, but there are others there. And some of them have more details that you don't need to study and will not be evaluated. So only the material covered in the lectures and the details covered in the lectures will be evaluated in the exams. So let's start then with the content we want to cover today. We'll do the best to cover this, but you know, don't get too stressed out. We do the best we can do, and there's no problem to continue covering this in the next lecture. So we will start with where we left before, that is, where is our immune system, and then we will talk about different aspects of antibody. Okay, so where is the immune system? We already covered this lecture. We said it's in the tissues, in the lymphoid organs, in the blood. For the innate immune system, the adaptive immune system is also in the blood and in the tissues, but we also said that it's in lymphoid organs. And so we learned that there are two types of, we also learned that the lymph is the plasma that leaks from the blood into the tissues. We already covered this, right? Is in the extracellular fluid, and is collected through the lymphatic vessels that are all throughout our body, okay? And the lymphoid organs are different organs that contain lymphocytes, that we will learn more about them today. They also contain other cell types that provide the structures and support and maintenance, and they are important for the maintenance and circulation of the lymphocytes. But the primary cellular component of the lymph are the lymphocytes, and that's where the lymphocyte names come from, because they are the main cellular component of the lymph. Okay, just anecdotically. Okay. So we learned that there are different types of lymphoid organs. The primary lymphoid organs, where the lymphocytes will develop from the immature progenitor. So we know that B cells and T cells developed from the common lymphoid progenitor that are in the bone marrow. But these common lymphoid progenitors will also migrate to the thymus to give rise to T cells. And they are named T cells because they are generated in the thymus, that start with T, and they will stay in the bone marrow, the common lymphoid progenitors, to generate B cells in the bone marrow, and that's why they are called B cells, because they are generated in the bone marrow. So when both thymus and bone marrow, where lymphocytes develops, are called the primary lymphoid organs, okay? And then we have the secondary lymphoid organs, where lymphocytes are as well, and is where mature naive lymphocytes resides, and also is where the adaptive immune responses can be initiated, okay? And here I have highlighted some of the secondary lymphoid organs, like, for example, the lymph nodes, and we will look at their structure, the spleen, the Peyer's patches that are part of the gut-associated lymphoid tissue that we will learn today about, and the tonsils that you know, many times we have to remove, but basically they are secondary lymphoid tissues. Okay, so we said that the naive lymphocytes reside in the lymphoid tissues. Maybe reside is not the best word. They recirculate in these secondary lymphoid tissues. So the naive lymphocytes continuously survive the secondary lymphoid organs for evidence of infection, okay? Same with the majority of memory lymphocytes, and we may have time to learn more about that in the memory lecture. Okay, so here we already learn as well that lymphocytes, this is here a lymphnode, okay, and we learn that the lymphocytes will arrive into, these are in yellow are all naive lymphocytes, right? Once they are generated from the thymus or bone marrow, they will get into the blood, and they will arrive into the lymphnode via the artery that vascularizes this lymphnode, okay? And they will then in the lymphnode, they will search for potential antigens that would engage them. Okay, if they didn't find any antigen, then they will leave these lymphonodes via the efferent lymphatic, okay? That's how they live, these yellow little lymphocytes. And then in these efferent lymphatics will continue through all the lymphatics in the body, and this may imply searching and surveying other lymph nodes that are part of this lymphatic system until they will reach the thoracic duct that will bind, turn the lymph into the circulation, specifically on the subclavian vein. And so the lymphocytes will then go into circulation, blood circulation again, and all the process will be starting again. And so all the time they do that. The naive are all the time traveling through lymph and blood. And in doing so, they are surveying all these secondary lymphoid organs in search for potential antigens that may clonally select them. If they are not clonally selected, they go and continue searching. And so at the same time, and this is again most of what we already said. So here you have naive lymphocytes that will arrive at the lymph node in the blood artery. Okay, so here is the lymph node. They will arrive via the blood artery, is what we just said a couple of seconds before. After that, they will search for their specific antigens, and they will leave and return to blood via the lymphatic system, and then will through the subclavian vein, it will get again into the bladder circulation. In the meantime, the lymph is bringing antigens and cells containing antigens from the tissues into the lymph node. So the pathogens from the site of infection reach the lymph node via the lymphatics. And so the lymphocytes are surveying all the time these nodes, okay, and secondary lymphoid tissues. And the lymph is bringing pathogen and pathogen-derived products into the very same lymph node or the draining lymph nodes from the site of infection, and establishing a meeting place for the lymphocytes and their pathogen. Is that clear?
I have a question.
Yes, please.
So when you're sick and your lymph nodes begin to swell, is that due to the clonal expansion of the naive lymphocytes?
Absolutely correct. You can start teaching now the lecture.
Okay.
Thank you. Thank you. Good question. Excellent question. So when it's inflamed, it's because there is clonal expansion. So there is more and more cells, and that's a good sign that we are responding right to the infection. That's because the lymph has carried the pathogen or pathogen antigen to the lymph nodes, and some of the lymphocytes specific for the invading pathogen are being clonally expanded. Okay, so then I think that you all understand now why the secondary lymphoid organs compartmentalize the infection and provide a meeting place for the cells of the adaptive immune response, the lymphocytes, to start and to encounter their specific antigens. This meeting place will enhance the chances for the lymphocytes to encounter their specific antigen. And so here is the anatomical structure of a lymph node. Like we will see for any secondary lymphoid tissues, there are some B-cell zones. B-cell zones here depicted here, sorry, and some T-cell zones here depicted in blue, okay? But they are very clear defined B-cell zones and T-cell zones, okay? We also learn that the T lymphocytes may arrive into these lymph nodes via the artery, right? The blood circulation. We just said that in the previous slide. Here is the artery that goes in. They may also arrive from an afferent lymphatic if they have already been in the lymphatic system. They will arrive from an afferent lymphatic. And they always leave through the efferent lymphatic vessel, okay? Okay, so the dendritic cells, we learned that they link innate and adaptive immunity because we learned that they have many pathogen recognition receptors that will engage the pathogen, will start producing cytokines, but also these dendritic cells now in the tissues are able to pick up antigens and migrate through the lymphatics vessels and get through the afferent lymphatic vessel into the lymph node. And you can see here is depicted how dendritic cells that have uptaken antigen will get through the afferent lymph into the lymph node, but also free antigen and free bacteria, right? Free pathogen can get into the lymph nodes as well through the lymph and the lymphatic vessels, all through the afferent lymphatic vessels. And in these lymphatic vessels, as in the tissues, we also have macrophages and we also have more dendritic cells. And so remember, the T cells or other lymph or B cells are coming and surveying all the time this lymph node, right? So they are coming through the artery or maybe coming through the afferent lymphatic. And so they will, then when they reach into the lymph node, they will start scanning these dendritic cells and see if they have, the dendritic cells have antigen for which their BCR, their TCR is specific, okay? The B cells, on the other hand, they don't need antigen presentation by the dendritic cells, but they may engage the antigen, free antigen in the lymph node. And so the lymphocytes that have engaged their specific antigen will be activated, will proliferate and differentiate, clonal expansion, right, into effector cells that could be effector CD4 T cells, effector cytotoxic CD8 T cells, or plasma cells that produce antibody. And these effector cells will leave they always leave through the efferent lymphatics, okay? And they will go into the tissues. And now they are effectors, they are empowered, and they can fight the infection in the tissue. Okay, any question about that? Okay, so the spleen deals with pathogens that make it to the blood. So the spleen is another secondary lymphoid tissue, and its anatomical organization and function can be divided into the white pulp of the spleen that is very similar to that of the lymph node, okay? And also the red pulp where the red blood cells are, that is not relevant for the initiation of or for response. The difference between the spleen and the lymph node, they are both secondary lymphoid tissues. They both have the B cell zones and the T cell zones. However, in contrast to the lymph node, both pathogens and lymphocytes enter the spleen through the blood, okay? The spleen does have no connection with the lymphatic system. So both the pathogen and the lymphocytes, they always reach the spleen via the blood. And so the spleen is very important to fight pathogens that have reached the circulation, the blood, okay? Like in a sepsis, for example, that the bacteria may reach the blood. And then we have other secondary lymphoid tissues that are the gut-associated lymphoid tissues. And so basically these are lymphoid, specialized immune system sites that are in our digestive tract. And you can see here they are underneath the epithelium of the gut. And again, as other secondary lymphoid tissues, they have B cell zones and T cell zones, here depicted in yellow and blue respectively. The difference here or uniqueness of the anatomy of the gut-associated lymphoid tissue respect to the spleen of the lymphoid node is that the route of the path entry is different and is direct, the antigen or the pathogen is directly delivered via these cells that are called M cells and phagocytic cells that basically can deliver antigen from the gut lumen directly into these lymphoid tissues, okay? And in these lymphoid tissues, there are dendritic cells that now can present this antigen to the T cells to initiate the immune response. So the other difference is that many times the migration pattern of lymphocytes after they have been activated in these gut-associated lymphoid tissues, they tend to stay within this mucosal system. So these activated T cells from the gut, they will recirculate and stay within the gut mucosa. And that makes sense because if the pathogen, they've been activated in the gut, right? The pathogen is probably a pathogen that is, the route of entry is oral. And so it makes sense that the T cells will stay in the gut to fight this pathogen that is cause infection through oral inoculation or oral contact. So let's learn more about antibodies now. So, which is, we already learned that, and this is, we even had a question on this. So how do we produce an infinite variety of antibodies? Each B cell expresses a unique antibody receptor that is selected by the antigen. So we already mentioned that each B cells express a unique type of antibody receptor. Of course, this is a simplification. There are many copies of the same BCR all throughout the plasma membrane of a B cell, right? But it's only one type with one specificity in each naive B cells, and the same is true for the TCR in the T cell. And so the B cell that again engage the specific antigen in the secondary lymphoid tissues will be clonally selected, activated, clonally selected, and proliferate, differentiate into effector B cell, which is a plasma cell that will produce antibodies. And a small proportion of these B cells and T cells that are activated will become memory cells that will be long-lived cells that will protect us from reinfection. And so the plasma cells, we already know this. They are effector B cells which secrete large volumes of antibodies. And that's why they have this massive endoplasmic reticulum here depicted in this cartoon, because they need to produce a lot of protein, okay? Okay, so let's learn more about the antibodies. We already covered some of these in the previous lecture. So we already learned that each antibody molecule will have 2 heavy chains and 2 identical light chains. So 2 identical heavy chains and 2 identical light chains. Each heavy light chain will have a variable region and a constant region. And each heavy chain will have a variable region of the heavy chains and a constant region of the heavy chains. And so the antigen binding site is formed by a combination of the variable region of the light chain and the variable region of the heavy chain. And so in each antibody molecule, how many antigen binding sites will we have? You can be wrong, just tell me a number.
Is it just 2?
Yes, 2 antigen binding sites, right? One here because we have two identical heavy chains and two identical light chains. And so now the heavy chains come in can have 5 different constant regions, and truly even there are variants within these 5 classes, okay? And so the 5 main classes are called immunoglobulin M, immunoglobulin G, immunoglobulin A, immunoglobulin D, and immunoglobulin E. And within the G, we have subclasses, 4 subclasses, and within A, we have 2 subclasses, okay? So a total of 9 different classes of immunoglobulin that basically what they have is different constant chain of the heavy chain, okay? Different constant region of the heavy chain. This part here, okay? The light chain can also have different constant region, is not biologically relevant for the function of the immunoglobulin, but it can have different regions. So there are 4 different subclasses or of the kappa constant region, the immunoglobulin kappa of the light chain, and 1 immunoglobulin lambda change, okay? So in total, there are 5 different constant regions of the light chain, this part here. So now the variable region of any of the 2 chains is what will determine the antigen binding site, and therefore will determine the antigen recognition, antigen binding, and the specificity of that particular antibody. And so there are millions of specificities and in our body, again, for antigens that we haven't seen yet. The constant region of the heavy chain, so this part, the constant region of the heavy chain, is what gives the antibody the biological activity, and we will learn more about that. Okay, so this is another cartoon to depict the same concepts. So we have here, each antibody has 2 identical heavy chains, 2 identical light chains. The variable region of the light chains and the variable region of the heavy chains will form the antigen binding sites. There are 2 identical antigen binding sites per each antibody molecule, okay? So these 2 antigen binding sites will bind exactly the same antigen. And then the antigenic determinants is the name that the part of the antigen that gets in contact with the antibody receives that name, antigenic determinants. Okay. The antigen binding site, as we said before, will determine the specificity of the antibody because it's the part that will bind to the antigen. And the constant region of the heavy chains will determine the biological activity of that antibody. For example, the capacity to activate complement, and we will learn more about that. So now scientists have worked with this, trying to understand the antibody structures for many years. And one experiment they did was to cleave the antibodies with an enzyme called papain. And so when they did that, they basically broke the antibody parts. 2 of the parts are identical, and they are called FAB because they contain the antigen binding sites, okay? So each FAB molecule has 1 antigen binding site and will have the light chain and part of the heavy chain. And then the other part that was resulting from this enzymatic cleavage of the antibody was the FC region, which contains most of the constant region of the heavy chain. And that's the FC region is what gives the antibody activity. And we will learn more about it. But that's a terminology that you need to know, because we will be referring to that. So FAB is called the fragment that has the antigen binding site and is the region of the antibody that contains the antigen binding site. The FC is called fragment crystallizable because it was easy to crystallize when they were doing the experiment, and is the region of the antibody that ensures that each antibody generates an appropriate immune response for a given antigen. So it will give the biological activity of to this antibody by binding to a specific classes of FC receptors and other immune molecules, such as complement, for example. These FC receptors are localized in innate cells, including macrophages. So remember when we were talking about the functional outcome of the B cells and the antibodies being able to neutralize, opsonize, and fix complement, this is all mediated through the FC portion of the antibody. These biological activities of the antibodies are mediated via the FC portion of the body. And the macrophages, for example, they will have FC receptors that will allow internalization of a bacteria via opsonization. So FC and FC receptor interactions facilitate antibody-mediated opsonization. Okay, any question about that?
So you were saying how protease cleavage by papain happened in the laboratory. So is
Yes.
papain circulating in our lymphatics and circulatory system to cleave it into FAB and FC?
No. I was never asked that question. That's very new, innovative mind. But no, not that I know. I never heard about that. Yeah, thank you. So the form in which antibodies will exist in nature is the full form, right? This is an experiment. It's not how antibodies exist. And it makes sense because you wanna have linked the antigen specificity and the biological activity in one molecule, so then the antibody can bring the pathogen to which it is specific into the macrophage that has the FC portion, right? If it were unlinked, it wouldn't serve the purpose, right? Makes sense, Sydney?
Yeah, I got it.
Okay, thank you. But I need to teach this, even if it is not physiological, because this terminology is used a lot, and especially you need to understand this to understand what are the FC receptors. Okay, so let's go with what are hypervariable loops or CDRs in the antibody molecule. So the hypervariable loops of the light change are represented here, and it has 3 hypervariable loops or complementary determining regions. And these complementary determining regions, and again, this is just in the light chain of the one light chain of the antibody. So these complementary determining regions are localized in the variable region of the antibodies and are structurally defined as the region of the antibody that is actually in contact with the antigen. So these will be the regions here that will be touching or be in contact with the antigen to which this antibody is specific. That is, they are amino acids in the polypeptide chains that formed the binding pocket for antigen and constitute these complementary determining regions that are basically the same as the hypervariable regions. So the CDRs, the complementary determining regions, are roughly synonymous equivalent to the hypervariable regions of the antibody. And so this is evidence that these hypervariable regions exist. So here the scientists, what they have done is they took all the antibodies in the serum of a person. And so this is in the x-axis here is the position of each amino acids in the heavy chain variable region and in the light chain variable region, okay? So here we are, is depicted. And so they plot in the y-axis the variability, okay? So in most of the molecules have very poor variability in certain regions, okay, that are called framework regions. You don't need to remember those names. But there were some regions that were called hypervariable regions where the amino acids, the residues among the different immunoglobulin molecules, the different heavy change variable region and the different light change variable region in all the antibodies molecules that they were analyzing was very different. So the sequence of amino acids in these regions were very different, and they were called hypervariable region. And it makes sense that it was different, right? We said that the antibodies in our body, they have millions of specificity, right? The BCR. And so this is the part of the antibody that is in contact with the antigen. So it's defining the specificity of the antibody. And if there are millions of different specificities, it makes sense that there is great variability in the amino acids that are forming these hypervariable regions that are in contact with the antigen. Okay. Within these three hypervariable regions, the hypervariable region 3 or the CDR3 has the highest variability. And for now, you just remember that and you will understand why in the next couple of lectures, okay? I will bring it up again. Okay, any question about that? Okay. So how and where does the antibody recognize the antigen? And we already learned what is epitope, right? Epitope is the part of the antigen that is recognized and bound by the antibody, okay? It's called also antigen determinant, okay? And we already learned that in contrast to T cells, the B cells, and like innate cells, they recognize the pathogens in their native form. So they don't need the antigens to be processed into peptides. They recognize the antigens in their native form. However, this needs to be, this epitope, which is the part of the antigens that is recognized by the antibody, needs to be accessible. Same thing is true for the TCR, okay? For the antibody needs to be accessible. The part that is same is true for the TCR is that also the epitope for the TCR is the part of the antigen that is being recognized by the TCR. So now, in the case of the antibody, because they recognize the antigen in their native form, this part needs to be accessible. And this epitope can be linear, or can be, so all the amino acids in the epitope are together, for example, in a protein antigen, or they can be separated, but they become in close proximity when in the 3D structure of the particular protein antigen, okay? So it can be linear epitopes or discontinuous epitopes, but they need to be accessible in their native form for the antibody to be able to bind to them. Okay, here is depicted the part of the proteins that are color white, and so this is a virion here, I think it's pox virus. The part of the proteins that are color white are the epitopes or part of the protein which the antibody, which is depicted here, will bind, okay? And again, the parts of the antibody that will bind to the antigen, which are called what? is the question for you. Anyone that wants to say which is the part of the antibody that will be in contact with the antigen?
The CDRs?
The CDRs or hypervariable regions. Good job, thank you, Serena. So the BCR antibody can recognize all type of chemical structures. So the T cells can only recognize peptides derived from proteins, but the BCR of antibody can recognize all types of chemical structures, including proteins, carbohydrates, glycoproteins, glycolipids, peptidoglycans, even metabolites, okay? So this is ferrous is a metabolite, for example, formed with ferrum, and
Professor, I think you muted yourself.
Okay, so it can be recognized here, the HIV peptide, the hen egg lysozyme is another protein in the eggs, the HIV GP120 protein, and you can see here how the variable region of the heavy change and variable region of the light change form this antigen binding site that may be different depending on the specificity of the antibody. So the binding between the antigen and the antibody is based in non-covalent forces, including electrostatic, hydrogen bond, van der Waals forces, and hydrophobic interactions that will be determining by the amino acids in the hypervariable region that are in contact with the antigen, as well as the amino acids on the antigen side that are part of the antigen determinants or the epitope in the antigen. Okay, so this came up this morning. You guys cannot complain that this is not an up-to-date course. This came up this morning. Yeah, here is published. I don't know, but it just came up in Nature. It was impressed in October 12th, but came up. At least I saw it in the Nature today. But it's very new article, okay? So, and it's called SARS coronavirus 2 neutralizing antibodies structures inform therapeutic strategies. And so in this, there has been a lot of interest in these neutralizing antibodies that are able to bind to the receptor binding domain of the ACE protein, which is the receptor for SARS coronavirus 2. Okay, and so here in gray is this particular part of the SARS coronavirus 2 spike protein, the receptor binding domain. This is the viral protein that will bind to the receptor, okay, in the cell. And so the receptor in the cell is not depicted here. But each of these ones are representative of different classes of antibody. And so in this paper, they have determine the structure of these antibodies that are capable of neutralizing the receptor, the viral protein, particularly and the receptor binding domain of this viral protein to prevent the SARS coronavirus 2 to get into the cells. And of course, there are great interest in that because they can be used for therapeutic purpose. And you have heard now in the news, the therapist, again, our president was injected with these antibodies to, some neutralizing antibodies to attenuate the spread of SARS coronavirus 2. Okay, there are different classes, and so some of these neutralizing antibodies bind to the upper part of the receptor binding domain. So you can see this also illustrate how they are highly specific. So this is just a part of the viral protein. It's the part of the viral protein that binds the receptor. And even within that part, some antibodies bind to the upper part, some binds to the lower part of the receptor binding domain of the viral glycoprotein, and some bind a little bit to both, right? And so they, again, this is important for therapeutics because there is a lot of interest in using these antibodies as passive immunization to contain the infection. Okay, so
I had a question.
yes,
So about the previous slide, so are like all those like C002, are those like different names of antibodies?
yes, they are basically how they name them, as they, I think that this may be the Regeneron, for example, you know, how they name them, the company or the investigators that reported them, right?
So
Not
did they, so are those classes like, are those like things that they divided all the antibodies into depending on where they bound on the ACE protein?
Where they bound on the on the viral protein. The ACE
on the
protein on our cells, right?
Yeah.
You got it, Larissa. Yes.
Okay. Cool.
It's pretty cool. Yes. Okay, so what is the difference between affinity and avidity of an antibody? So the antibodies, we already said, come in 5 flavors, and they may be subclasses within IgG and IgA, but 5 general flavors: IgG, IgM, IgD, IgA, and IgE. So these are the immunoglobulin isotypes or classes, and refers to the genetic variations in the constant regions of the heavy chain, and in the case of there are isotypes also for the variable region, for the constant region of the light chains that we mentioned before for lambda and kappa. However, only differences in the heavy chain constant region defines the 5 main classes of immunoglobulins. So what is relevant for the isotype is the heavy chain of the constant region. Okay? The constant region of the heavy chain. Okay? Is that clear? I could say it again. Okay, I think we said it before too. So which are the mechanisms for generating different isotypes? And so a biological mechanism that changes B cells' productions of antibodies from one isotype to another isotype is called isotype switching, okay? So a B cell always starts producing IgM, and then can switch to other isotypes, and that process is called isotype switching. This isotype switching can be used to change the isotype of a heavy chain, but not the light chain, okay? So the light chain constant region doesn't change via isotype switching, only the isotype of the heavy chain. And that will change the biological activity of the antibody, because it's changing the isotype that is determined by the constant region of the heavy chain, okay? That determines, again, the Fc portion that determines the biological activity. However, the isotype switching does not change the variable region. So the isotype switching will not change the antibody specificity for a particular antigen, okay? Is that clear? Okay. And this process, the isotype switching, always requires CD4 T cell help for the B cell, okay? So that for the B cells to change the isotype, they need to receive help from the CD4 T cells. And remember, in the previous lectures, we mentioned that the T follicular helper cells, a subset of CD4 T cells, is the one that can help the B cells, okay? And we will learn more about this later in the course, but just to start introducing it. So the constant region takes the function and structure of the antibody. IgM is the first one to be produced during an immune response. During B cell development, IgM is the first one to be expressed, and IgM is the exclusive isotype expressed in immature B cells. And we will learn in the next couple of lectures where are these immature B cells. And so IgM is produced early on, and it does not require, because it's the first one to be produced, it does not require T cell help. The T cell help is necessary for changing into another isotype, from IgM into another isotype, or any change in the isotype. But because this is the first one to be produced, it can be produced without T cell help. It also have low binding affinity, and we will learn later why that makes sense, that it has low binding affinity. And what is the affinity? Is the strength with which the antigen and the antibody binds to its specific antigen. Okay, so each antibody molecule, each IgM will have lower affinity. However, this is compensated by forming pentameric IgM molecules via this J chain that bring 5 individual IgM molecules all together in a pentamer. And that's how IgM exists in circulation. And so while the affinity, strength to which each IgM molecule binds to the antigen is very low, the strength combined for all these antibody that are brought together, these 5 IgM that are brought together, that's much higher, okay? And that's the difference between avidity and affinity. So affinity is the strength of interaction between one antibody binding site. So here, one antibody binding site in one FAB, okay? And one corresponding epitope on its antigen, okay? So this is the affinity. Now, the avidity is the combined strength of binding of multiple sites of antibody. For example, IgG, which is monomeric always, will have 2 antigen binding sites. So its avidity is the strength of the 2 antibody binding antigen binding sites combined, okay? And for the IgM, is the avidity is the strength of their 10 antigen binding sites combined, because it's produced in a pentameric form, okay? So the avidity is the combined strength of binding of multiple sites of an antibody. Okay, so the immunoglobulin isotype and affinity can change over the course of the response. So here is the time days after an antigen is inoculated. So in the primary response, the first time this antigen is seen, the first one to be produced early on is IgM, as we just mentioned, is the first one always to be produced. And it has low affinity. And later, there is via CD4 help, we will have isotype switching into IgG that has higher affinity. And then we will learn much later how in the secondary response, we already have some IgG that is left over from the primary response and will be of higher affinity. And also we have B cells that have already undergone the switch, and they will start producing IgG right after secondary encounter with antigen. So the IgM is ideal for complement activation due to its pentameric.
Professor, you're muted again.
IgM exists in this planar form and when it binds to the pathogen, it acquires this stable form, and this is perfect for combining and attaching the complement factor C1, which is also pentameric. Okay, so it's a perfect fit, and that's why it's so good IgM at activating complement. So IgG is the most abundant antibody isotype found in the circulation. And so 75% of the serum immunoglobulins in our serum is IgG. And as I mentioned before, there are 4 subclasses of IgG, IgG1, 2, 3, and 4. And they are named based on the abundance in the serum. So IgG1 is the most abundant in the serum. IgG4 is less concentrated in our serum. And these IgGs are key immunoglobulins for vaccination because they are good at neutralization, opsonization, and complement activation, with some variations among the subclasses that we will not go into details. So here is depicted, for example, how repeated injection with an attenuated virus will induce IgG production that will have high affinity and will neutralize very well this virus. And this protection for some viruses can be up to 10 years or more. Okay, so that's why when you get vaccinated, for some vaccines, we don't need to have a repetition of the vaccination because the IgG can protect us for many years. Here is depicted how, for example, IgG against the bacteria will favor opsonization of the bacteria. So then we have IgA. IgA is a monomeric, it can be monomeric in the serum, but more importantly, IgA is produced in a dimeric form in the mucosal surfaces. And this dimeric form has the capacity to cross the epithelial. And that's why it's so important for mucosal surfaces, and it's also very high in the breast milk. So when the baby drinks the breast milk, the IgA will protect the baby from the commensals or outgrowth of commensal in the gut, for example, and potential dysbiosis that could cause inflammation or disease. And IgA is also very good at neutralizing antigens, okay? So IgA can export toxins and pathogens from the lamina propria. So this is here the epithelium in the mucosal. So it can export toxins from the lamina propria underneath the epithelium into the lumen, okay? So get rid of these toxins. It can also bind and neutralize antigens in the lumen and internalize them in the epithelial cells or the M cells for presentation by degradation in the epithelial cells, or for presentation of by the dendritic cells into the in the gut-associated lymphoid tissues, and subsequent activation of T cells. So the secreted IgA in the gut surfaces, or in the case of the baby, the IgA that is drunk via the milk, right, can bind and neutralize pathogen and toxins in the lumen of the gut. And then we already mentioned this, how it can pick up antigens either in the lumen or in the endosome of M cells and bring it to the lamina propria for the dendritic cells to use them to present antigens to the T cells, as we learned here that was important for the gut and activation of T cells in the gut-associated lymphoid tissue. Okay, so let's see what time is it. I want to recommend you to watch this global immuno talk. Again, it's very advanced. You can do it now, you can do it after the course ends, but it's very interesting. If we have time, I will project a little bit of it, but if not, you can go to YouTube, and it's talking about IgA producing cells in health and disease, and includes also IgA production in SARS coronavirus 2 infection. So IgE finally is, and we will learn more about IgE, but it's 3,000-fold less than other isotypes in concentration, so it's very low concentration. It has the shortest half-life when soluble, but long when bound to its Fc epsilon receptor. And what is interesting is that in most cases, the Fc receptor can bind the immunoglobulin only when it is accomplished to an antigen. But IgE is an exception. It can be bound to the Fc receptor even when it hasn't yet been accomplished to the antigen. And we will learn more about that, but the IgE, the Fc epsilon receptor, so the receptor is specific for the constant region of the immunoglobulin E, are present in mast cells, in eosinophils, and in basophils. And once they are cross-linked, they produce histamines and lipid mediators that promote muscle contraction, and they also produce secretion of cytokines. And this is important to fight these big parasites, the helminths, that cannot be uptaken by the phagocytes, but instead they may need to be eliminated by muscle contraction and peristaltic movement of the gut that eliminate these big parasites, okay? Also, the eosinophils will release some proteins that will break the membrane of these big parasites after IgE cross-linking. Sorry, IgE epsilon receptor cross-linking. So the IgE against harmless antigens like pollen, for example, is what causes allergies, okay? And we will have a lecture about this. So I would like to summarize here a little bit of what we learned about the isotypes. So different antibodies isotypes are found in different parts of the body and serve different functions. And so, for example, here is depicted where the different antibodies isotypes are in our body. IgM, for example, is very big, right? We said it exists in a pentameric form. And it is not easy to extravasate this big IgM, so it makes sense that it's mostly in the circulation within the vessel, the circulatory vessels, okay? So that's why it's here depicted in the heart. So IgG is very interesting, specifically IgG1 can be transported via the placenta, so that's why IgE is here in the baby, okay? And also, IgG exists in a monomeric form. It's always monomeric, so it's really good at the extravasate. And that's why, like the monomeric IgA, they will, again, be good at diffusion into extravascular sites, and they will be present in the tissues, okay? In all our tissues, once they are produced. And as I mentioned, IgM is really bad as extravasation, and that's why it will be mostly in circulation. And then finally, we already know that dimeric IgA is really good at crossing epithelium, and that's why it's really good and is present in the mucosal surfaces. And so what are the functions? We already learned that IgM is really good for activating complement. We already learned that we really want to have IgG to protect us from pathogen, and vaccination triggers IgG in many cases, also IgA, and because it's good for ozonization. Many of them are good for neutralization, and also with different variability among the subclasses, they are also good for complement activation. We also learned that IgE is very low in concentration, as depicted here, much lower than other isotypes, and is good for sensitizing mast cells, eosinophils, and basophils that are important for fighting helminths, big parasites, and also cause allergies. Okay, so each response will generate multiple isotypes, okay? Each time we see an antigen, will generate multiple isotypes derived from different B cells that have different specificities, okay? So, okay, so I want to end with the summary. Where is our immune system? We learn about primary lymphoid organs, the bone marrow and the thymus that are important for development of lymphocytes, the secondary lymphoid organs that are the lymph nodes, the spleens, and the gut-associated lymphoid tissues, which provide a meeting place with B cell zones and T cell zones. And we learned that the antigen arrival to the secondary lymphoid tissues may occur in different ways. So we learned about the general role of antibodies, that they have a variable light change, a variable region in the heavy change, a constant region in the light change, a constant region in the heavy change, and which are the functions of that, the antigen binding or the biological activity. We learn about the FC portion that gives the biological activity to antibody, and FAB portion that gives the antigen binding specificity to the antibody. We learn what are the hypervariable loops or the CDRs, that are the parts of the antibody that are in contact with the antigen. And we learned that on the other side, the epitopes are the parts of the antigens that are bound by the antibody or the TCR. We learned that the affinity is the strength of one antibody binding site, while the avidity is the strength combined with by multiple antibody binding sites. And then we learn about specific functions and localization of the different 5 different isotypes of immunoglobulins. Okay, so with that, we'll end the lecture.